For every 3 people in the world, 1 is infected with tuberculosis. Diagnosing active tuberculosis prevents new infections because patients with active tuberculosis will go on to infect between 10 and 15 other people per year until the time when they are either treated or die.
MODS is an acronym that we use to avoid having to say microscopic-observation drug-susceptibility assay, and the reason that it's called that will become apparent as the methodology is explained. In this short video, we will outline the MODS methodology for the rapid, inexpensive diagnosis of tuberculosis and multidrug-resistant tuberculosis. The video will set out the methodology step by step. We'll discuss the biosafety implications of working with liquid culture, and we'll also explain some of the important quality assurance issues that are required for use of this methodology. The MODS assay was developed at Cayetano University in Lima, Peru, in the laboratory of Bob Gilman, following observations initially made by Luz Caviedes. Luz was trying to identify rapid ways of detecting mycobacterium tuberculosis in cultures using colorimetric indicators. But she noted that the observation of those cultures under the microscope revealed the presence of growth of mycobacteria several days before the colorimetric indicators changed colour, and it was based on that observation that the MODS assay was developed. The MODS assay was developed in a resource-limited setting and is targeted at resource-limited settings. Therefore, at just under $3